During protein synthesis, the living cells are trying to make proteins to make parts of the body, such as red and white blood cells. Protein synthesis is divided into transcription and translation. Proteins are made in the ribosome, and they're made of one amino acid, one codon, and three bases. Transcription, the actual synthesis of mRNA, occurs in the direction of DNA. The purpose of transcription is to create messenger RNA for DNA, and there are three steps of transcription. First step is initiation. The promoter region is where the TATA box is located. The promoter region is found upstream of the DNA. The transcription factors then bind to the TATA box and the RNA polymerase, then attach to the promoter region, going down the template strand. This is the beginning of RNA synthesis. Of transcription. This is where the RNA polymerase 2 runs through the DNA strands and unwinds the double helix. It elongates the RNA into 3 to 5, and then from there, the DNA is able to reform its double helix. The sense strand, or the template strand, carries the information that will be transcribed. The antisense DNA strand goes through translation and forms a protein. The RNA polymerase creates mRNA in a 3 to 5 direction anti-parallel to the original mRNA. The 5 to 3 orientation of mRNA is due to the anti-parallel of DNA. It's orientated this way because RNA polymerase can only add bases in this direction. The next step is termination of transcription. This is when the RNA polymerase 2 reaches the stop region in the DNA strand. From there, it continues 10 to 30 bases down the DNA until it reaches a sequence of A, U, A, and releases pre-mRNA. From there, the RNA polymerase detaches from the DNA and the pre-mRNA is freed from the DNA strand.
Processing happens for the transcript leaves nucleus by adding a 5' guanine cap and a 3' poly tail to the ends, which protect the transcript from enzymes as it leaves the nucleus, and it allows ribosomes to attach the 5' guanine cap. mRNA splicing is the act of removing introns, which are non-coding segments, from the mRNA before translation, leaving only the exons, which are the coding segments for amino acids. It's done so by getting protein snRNPs and other proteins to create a spliceosome, which then pushes on the intron on both ends and pushes it out into a cutout intron, leaving only the exons to create an mRNA, which is then translated. The mRNA coding dictionary is what tells you what codons code for which amino acids. It works as you get the first, second, and third letter, and you follow it through the chart to determine what amino acid it is. There is 4 amino acids and codons that you need to remember. They're UAA, UAG, and UGA, which are the stop codons. And then you have AUG, which is the codon which codes for MET, which is the start codon.
The overview of translation. Translation is the making of the polypeptide chain. There are 3 stages to translation: initiation, elongation, and translocation.
Felicitate the specific coupling of mRNA and tRNA. mRNA is the codons and tRNA is the anticodons. Each ribosome consists of a small subunit and a large subunit. These subunits are made of RNA, ribosomal RNA, and other proteins, and made by RNA polymerase 1. Each ribosome also contains 3 binding sites of tRNA, which are the E, P, and A. The P site is responsible for holding the polypeptide chain. The A site is responsible for adding amino acids, and the E site is what exits the tRNA after it's done being added.
This is the tRNA structure. Here we have the tRNA molecule and the structure. The tRNA molecule is made during RNA polymerase 3. As you can see, it has 80 nucleotides, which clumple into 3 different sections. Here we have the anticodon and the attachment site, which is at the 3', and here are the amino acid attachments. This is amino acid tRNA synthetase. This is a special enzyme which is within the cytosol. There's 20 different types of amino acid tRNA synthetase because there's 20 different amino acids. So as you can see here, the amino acid comes in with energy, same with the tRNA. The special enzyme then binds the 2 together with the energy and then sends it out, and this leads it to the ribosome. This is the initiation of of translation. So here we have an mRNA strand. It starts at the five. Goes from 5 to the 3 end, and it's connected to the small ribosomal subunit. Here is a tRNA that comes down with its UAC anticodon and it connects to the start codon. It is carrying a methionine amino acid. Then, with the addition of energy, the large ribosomal unit becomes, and this finishes the initiation complex. And then this leads to elongation because this tRNA is in the P site, which leaves the A site open for elongation. This is the elongation and translocation of translation. So starting here, this is the process after initiation. We have a tRNA with polypeptides in the P site. So with elongation and translocation, a new tRNA comes into the open A site. Here, then the next step, the polypeptide chain breaks and connects to the A site. Finally, the tRNA in the A site will move to the P site with the polypeptide chain, and the P site will move to the E site and then leave the ribosome. This can occur many times and is a 3-stage process. The actual movement of the tRNAs is translocation, where the elongating of the polypeptide chains is the elongation. And this occurs very fastly. It occurs in 1/10 of a second. This is a termination of translation. This occurs after elongation. What happens is the ribosome hits a stop codon. This stop codon is either UAG, UAA, or AUG. When this occurs, the A site allows in a release factor, which is purple as seen here. This defranchises the tRNA and the polypeptide. This breaks them apart and separates them, sending them out. And then eventually both ribosomal subunits and the mRNA are broken apart and ready to be reused later.
Acid structure. So amino acids consist of 4 main parts, and that is the primary carbon, the amino group, carboxyl group, and the side chain, or R. And the R group is what determines the difference between amino acids and like which of the 20 it will be. RNA in a large ribosomal subunit acts as an enzyme to bond two amino acids together. The peptide bond is formed through dehydration synthesis, and it's called this because as the bond is formed, water is removed, and this bond occurs between the carboxyl group of one amino acid and the amino group of the other. Okay, polyribosomes are a cluster of ribosomes on a single mRNA molecule, and they simultaneously translate this mRNA to produce polypeptides. And this is a very efficient and quick way to produce them. Levels of protein structure. So there are 4 levels: primary structure, secondary structure, tertiary structure, and quaternary structure. The primary structure consists of the sequence of amino acids and a polypeptide chain, which is determined by DNA. The secondary structure consists of loops, sheets, and coils, bonded by hydrogen bonds between non-adjacent carboxyl and amino groups. The tertiary structure is a 3-dimensional shape or form, which is held together by the interaction of R groups and water through electrical charges. And the quaternary structure is made up of individual clusters of peptide and protein chains, which form a final shape. Post-translational modifications of protein. So after translation, polypeptide chains undergo modifications in the Golgi apparatus. Sugars, lipids, and phosphates may be added onto some amino acids. Other amino acids may also be removed. Proteins also undergo modifications that can affect the 3-dimensional shape.